Good afternoon, ladies and gentlemen, and welcome back to the afternoon session for this Crafoord Prize Symposium in Polyarthritis 2021. It's my pleasure to chair the afternoon session together with Professor Anna Blum from Lund University and also Professor Richard Holmdom from Karolinska Institute. My name is Qiang Hammarström from Karolinska Institute. So we have a number of exciting talks this afternoon, and I will introduce the first speaker, Dr. Yanick, who is online joining us from Paris or Edinburgh.
Hi.
Nice meeting you there. So Dr. Yanick has made seminal contributions to a group of autoinflammatory disorders that we haven't had chance to hear a lot yesterday and today. It's a group disorders called type 1 interferonopathy. So disease causing by mutations in genes regulating the type 1 interferon signaling pathways. So we're very much looking forward to your talk today, and the screen is yours, Dr. Crow.
Thank you. Thanks very much for the invitation to talk. I'm sorry not to be there in person, but I'm definitely there with you in spirit. And congratulations to Dan. Very well deserved and an absolutely fantastic accolade. And actually, it's not the first time. So Dan's a serial collector of prizes, and this is the last time that I was involved in this kind of thing with Dan in 2019 when he received the Ross Prize. And as a friend said to me recently, Yanick, you're always the bridesmaid and never the bride. But it's a real pleasure to be the kind of supporting act. So like Dan, I'm very interested in humans. And this is a quote from Sidney Brenner, who won the Nobel Prize in 2002 for his work on worms. And this isn't a quote, him sort of discussing with his Cambridge mates in the pub how it was that everybody at the LMB won Nobel Prizes. This is actually about the fact that the human can be your model organism. We're interested in using human disease to find out stuff about biology and a way, Mendelian disease in particular, as a way of deconvoluting complex biology. And the diseases I'm going to be speaking about to you today are these type 1 interferonopathies. So here's a definition. Monogenic disorders. I'm a rare diseases doctor. I'm a clinical geneticist in which there's an upregulation of type 1 interferon signaling. But I'm personally not very interested in upregulation of type 1 interferon signaling simply as a biomarker. I mean, that can be useful. But the idea here is that the interferon is actually contributing to the pathology. And why that is important is because if that is the case, then the implication of that statement is that blocking type 1 interferon signaling, reducing the amount of interferon being produced or blocking the signaling downstream of that interferon stimulus could be beneficial in the clinical context. Okay, so as far as I'm concerned, these type 1 interferonopathies are monogenic disorders because you can't really get much more innate than the type 1 interferon signaling system. But it is of note that almost every cell in the body can respond to type 1 interferon. So it's not necessarily that we're limited in this case to the same kind of cells that maybe Dan's work has highlighted. Okay, so type 1 interferon mediated monogenic autoinflammation. And what this really boils down to is a fundamental biological question, which is the differentiation of self from non-self. And as you'll hear later on from Jean-Laurent, COVID is just one of the most recent examples, if you will, of the war to the death that we fight as humans with pathogens. And if you cannot fight that war successfully, you lose and you die. And over evolutionary time, it's come to pass that the way we recognize, to a large extent, the way we recognize viral infection is by the recognition of viral nucleic acids, viral DNA and RNA. And when we recognize those nucleic acid species, what happens is there's an induction of this fundamentally powerful molecule, these type 1 interferons, and they set up an antiviral state. And that then involves a conversation with the adaptive immune system as well. But there's this first pass, absolutely crucial stage where you produce interferon in response to viral nucleic acid. And that, all told, that's a pretty good way of fighting infection. We haven't done badly. We come across situations where we're not so good at it, but it's not so bad. However, it begs an absolutely fundamental biological conundrum at the same time, which is that our own cells are replete with our own nucleic acid. So our cells are full of our own DNA and our RNA. And essentially, what we're saying in regards to these type 1 interferon-related disorders is that you have a misrepresentation of your self-nucleic acids as those of a virus. And so you induce an antiviral response against self. And that's really the size of it. And I could kind of stop there. So it's all about a case of mistaken identity, a failure of this absolutely fundamental principle of self, non-self discrimination at the level of the innate immune system. Okay, so and this is an idea that Jean-Laurent encouraged me to think about and write a little piece in 2011, and we did that. But in fact, as I've said to say many times, I've never actually had an original thought in my life, and this was all considered and mapped out by this wonderful man, Qiang, a virologist, an American working in Paris in the 70s, 80s, 90s. And Qiang in 1980 wrote this wonderful piece of literature where, as you can see in the red text box, he on the basis of the work that he'd already done, he said that one might expect to find instances in which too much interferon, instead of being beneficial. So this was when interferon had recently been identified and produced, and people were very excited about the use of interferon as an antiviral agent, perhaps as an anti-cancer agent. But he was saying, be careful, because the work that he'd done in mice and rats had suggested that sometimes interferon could be harmful. And he said that this was the subject of the presentation in his paper. And essentially, this is the subject of the presentation I'm giving today in celebration of Dan's work. Okay, so if this is such a big deal, you might ask, well, how come it was only in 2011 that this sort of idea was floated? And I think there's a very simple answer to that question, and it is that there is no routine assay in clinical medicine where we can say, as clinicians, does this patient demonstrate an upregulation of type 1 interferon signaling? And more generally, I'd make the point that I don't think that we use transcriptomics. Well, we don't use that routinely in medicine. And I think sometimes we really are missing a trick. And I guess as these things become more available and cheaper, basically, that may well come to pass. But there is no routine assay in clinical medicine. That's sort of becoming. I think that will become available, and in certain places, it is coming to pass. But essentially, it's not the case in most places. And the second point I'd like to make is that a priori, it's not completely obvious what the clinical consequences would be of too much interferon signaling. So what would the phenotypes be? And we had some idea maybe there might be something related to lupus and and one of the diseases we've been studying, Aicardi-Goutier syndrome, gave us some insights. But when I first wrote that little piece in 2011, it wasn't clear the breadth of the phenotype, if there was going to be any breadth, what the breadth of that phenotype would look like, that phenotypic spectrum would look like. So what we needed to do and what we did is develop some assays. So if there aren't any routine assays in clinical medicine, we needed to develop some, and we stole an idea out of the lupus doctor's book and developed this little interferon panel of so-called interferon stimulated genes. So there we're looking at the consequence of a type 1 interferon signal. And then we also developed a digital ELISA assay for measuring interferon alpha protein per se. And essentially, interferon is such a powerful molecule that the quantities, the concentration of at least interferon alpha in blood and cerebrospinal fluid and other tissues is so low that it's below the level of detection of routine ELISAs. So using this technology called single molecule array, we were able to measure interferon alpha protein at very low concentrations. But just because it's very low doesn't mean to say it's not having an effect. It's just it's extremely potent. And so armed with these two assays, we went looking for patients and looking for genes. So I'm just going to sort of quickly take you through this because it is our, it's our weapon of choice, if you like. Here we're looking at the consequences of a type 1 interferon signal, and here we have 6 interferon stimulated genes. You could use, well, now we use 24, you could do less, you could do more. But this is a young woman with a STING gain of function mutation, the classical V155M mutation in STING, causing this thing called SAVI. And here, just at the bottom left of each set of graphs, you can see the data for 29 control individuals. So look at the RQ values, look at the effect that this is happening. It doesn't matter when we take a blood sample from this young person. She is living in a chronic antiviral state. This is what happens when you have a virus, but there isn't a virus. It's as if she's being chronically infected, but there isn't an infection. It's because her immune system is triggering as if there is a virus, but there isn't one. And here we're using the actual measure of the ligand, the interferon alpha protein, and I've mentioned lupus very briefly. In dermatomyositis, you can see the same kind of thing. But what I want to draw your attention to here is if you measure the concentration of interferon alpha protein in the cerebrospinal fluid of people who have a viral encephalitis, for example, you get these kinds of concentrations of interferon alpha protein. And if you measure the cerebrospinal fluid, the amount of interferon alpha in the cerebrospinal fluid of patients with this monogenic interferonopathy called Aicardi-Goutier syndrome, you see the equivalent kind of levels of interferon alpha as you do in an infection, really highlighting this issue that it's as if these people are living as if they were chronically infected. And I just want to say now that interferon, as Ian Gresser had pointed out, is a toxic substance, and that's why I think it's kept at very low concentrations, except when you need it as a disinfectant. Now, the kind of USP of our lab has been that we haven't been too bothered about the phenotype. I mean, we're very interested in phenotypes and we're very interested in detailing them. But because we were kind of uncertain of the breadth of the phenotype that might be associated with too much interferon, we've been very agnostic, very promiscuous in terms of the number of people, the types of clinical phenotypes that we've screened. So what we do, is, a phenotype, not necessarily that bothered about the phenotype, or at least we weren't initially. We've learned the kind of phenotypes that are relevant. We're learning that. And we'll do the interferon screen using our ISGs or our interferon alpha protein assay. And if there is a persistent upregulation of type 1 interferon signaling, we would then go on to gene sequencing. And in this way, you learn stuff. You find out interesting things that perhaps might have been overlooked. And I'll just give you three very brief examples of that. So mutations in PSMB8 were first described in 2010, but it wasn't until 2012 when people cottoned on to the fact that patients with mutations in PSMB8 actually demonstrated an interferon signal. The same for this disease called copper, which I will talk about briefly in a moment. So copper was first described in 2018, but it wasn't until 2015, but it wasn't until 2018 until anyone actually looked at the transcriptome in these individuals and found that there was an upregulation of type 1 interferon signaling. And the same for this mitochondrial gene. So the mutations in this gene were described in 2016, and it wasn't until 5 years after that that we described mutations in ATA3A, those same kind of mutations, to be associated with an upregulation of interferon signaling. So we learned something about the biology that wasn't otherwise recognized, even though the genes had been identified some time before and studied quite carefully. So fast forwarding to 2021, although actually that's going backwards now, what I'd say to you is that you can make a case for mutations in any of these colored lozenges, so that when you get mutations in any of these genotypes, essentially, you can argue that the consequence is a type 1 interferonopathy. And you can jiggle those gene names about, and you start to see some sense. So the large majority of those genotypes, of those genes, code for proteins and or are involved in the metabolism of nucleic acid, or nucleic acid sensing, or nucleic acid signaling, or turning off a type 1 interferon signal. And when we say nucleic acid metabolism, essentially what I'm saying is that they do stuff to DNA or RNA, and in some sense they kind of, this is all about, remember what I said earlier on, this is about a misrepresentation of self as non-self. And so essentially what we're saying here is that these genes do something to self-nucleic acid, or the way that nucleic acids are sensed or signaled, to minimize the likelihood that you will misinterpret your own nucleic acid as that of a virus. And when you've got mutations in those genes, when you've got Mendelian mutations in those genes, you get this confused antiviral state against self. So we can sort of put a little bit more detail into that. So, one thing that you could do is to say, well, here's some of my DNA, RNA, but it's me. And so I'm going to put a label on it that says self. I'm going to frank it like a stamp, which says self. And I think that mutations in this double-stranded RNA editing enzyme, ADA, are an example of that, where you, by editing double-stranded RNA, you mark it as self. You might just have waste disposal systems. So, TREX1 is a DNase, which I think effectively chops up DNA that's sort of surplus to requirements, and acts as a waste disposal system. So you get rid of it in a controlled fashion, and it's not left in the cell to induce a type 1 interferon response. You may have threshold levels. So you might appreciate that you're going to have a bit of background nucleic acid that's coming from self, but at some point you'll decide that beyond a certain point, then things are going wrong. That it's more than self. That there's a virus involved. And if you have gain of function mutations, for example, in MDA5 or IFH1 coding for MDA5, this double-stranded cytosolic RNA sensor, then the sensor becomes twitchy and it responds to a much lower dose of background than it would otherwise. You may just keep. So if you're going to sense nucleic acids, sense viral nucleic acids, you may have a system where you compartmentalize the receptors, the sensors, or you keep, your stash of self-nucleic acid in a particular safe place, a safe haven, for example, within mitochondria or within the nucleus per se. And then you may have mutations involved in the downstream signaling, and things might go wrong there. Or, and I mentioned this earlier, that you may have mutations in genes that switch off an innate immune response. So it's very important to be able to kill viruses, but it's also very important once you've done that killing to stop, to switch the system off. And mutations in ISG15, for example, found by Jean-Laurent's group, or USP18, or a recent separation of function mutation in STAT2 are all examples of that phenomenon. Okay, so what about phenotypes? So I came at all of this initially from the perspective of somebody who was very interested in Mendelian neurogenetics. And what we became pretty interested in is the presence of intracranial calcification. So there are many, many, many causes of intracranial calcification, but it's clear that where you see intracranial calcification, it's a good marker. You should think about the possibility that there's an upregulation of type 1 interferon signaling. And a large number of these genotypes, that I've mentioned, are associated with this presence of calcium, which is best seen on a CT scan. So these are all CT scans, and is easily missed on an MRI scan. And if you're going to do MRI scans looking for this kind of thing, you need to use special sequences. And I just point out that although mutations in STING or TMEM173 are causing SAVI, essentially are not considered to cause neurological problems or be associated with neurological involvement, you can get intracranial calcification in SAVI. So, it's well worth thinking about this as a useful diagnostic clue. The second system, if I had to kind of synthesize this and be, make everything pretty simple and say, what are the systems involved most of the time in these type 1 interferonopathies, it would be brain and skin. And so here are some examples of the kind of skin lesions that one sees. And you get these, you get frank loss of tissue. You see here this young woman with a ACP5 mutation has lost an index finger. So this child's dead, this young woman's dead. This is a boy with STING SAVI mutation who's been treated with ruxolitinib, who's not dead. These are his cheeks when he was a little baby. And here's a picture from Rafaela Gobak-Mensky's New England Journal paper from 2014. And this is a woman with a D18N dominant negative mutation in TREX1. And, I'm not a dermatologist, but I have to say that looks rather like that. And that looks rather like that. And so what I'm wanting to say to you, I've mentioned already the fact that when we look at the genotypes, when we look backwards at the genotypes that have been identified associated with too much interferon signaling, there's a pattern there. They're involved in nucleic acid metabolism, interferon signaling. Also, when you look at these genotypes and look at the phenotypes associated with mutations in those genes, you see commonality. So this is AGS, this is TRAP, this is ISG15. Sorry, there's something with my slides here. But these are disparate disorders. These would not necessarily be put in the same diagnostic stable, but you can see that the phenotypic overlap, I think, is telling us something. And what I think it's telling us is that there's a common factor involved in the pathology. And I would suggest that the common factor in the pathology is interferon. So this is a prize for Dan to celebrate joint disease. So I thought I'd put a little bit of joint disease in, and I show you some interesting things that have come up. So this is gain of function mutations in MDA5 or AFH1. I've mentioned it already. Can cause a classical neuroinflammatory disorder called Aicardi-Goudis syndrome, but it can also cause other things. And in fact, so there is this entity called Singleton-Merten syndrome. I think it's all part of the same spectrum. I just point out that what they have is this deforming arthropathy. They can have this loss of tissue around the ears, for example, loss of teeth. They can get psoriatic type problems. And they can get intracranial calcification. So here's another example of that same phenotype or the same genotype. These are the hands and feet of a young woman from Brazil. These are the hands and the feet of her mother, and they both have a gain of function mutation in MDA5. So this is a severe arthropathy, but the joints are essentially reasonably pristine, actually. So there's a deforming arthropathy. And I think this is actually something to do with the fascia. And one other thing I want to point out in terms of sort of rheumatological joint disease is you can get insertion, you get calcification at the insertion of tendons, and you can get tendon rupture. So these people frequently get tendon rupture. So you can see again the calcification there. And the thing they die of is calcification of the cardiac valves. Okay, so there's some pictures of some X-rays of some hands, and then I'm not going to talk more about this genotype and this disease, but I just want to show you some more hands, and I want to say to you that mutations in DNase2 cause this kind of hand deficit, and these are the X-rays, and mutations in MDA5, as I mentioned already, cause this kind of hand deficit, and here are the X-rays, and I think essentially they're the same thing. And we also see the same kind of thing sometimes in patients with STING gain-of-function mutations. And all of that is somewhat reminiscent of this rather enigmatic entity, I think, called Jacquard's arthropathy. And effectively, I think that these are examples of, these are effectively monogenic Mendelian mimics of Jacquard's arthropathy. And so maybe that's telling us something about Jacquard's arthropathy, which is associated with lupus and psoriasis. Okay, so a little bit more joint disease because there's a theme to the meeting. And I'm just going to briefly touch on this disorder called Copper syndrome. So Copper syndrome was first described in 2015, and essentially it's a combination of joint disease and lung disease and kidney disease associated with autoantibody production. And I was very interested in this disorder because as soon as I learned about this disease, I thought that maybe it was a kind of, there might be some overlap with Savy syndrome, which is STING gain of function. So we went looking for patients with mutations in copper, and I'm going to tell you a very brief story. So here's an 11-year-old girl presented at age 2.5 years with joint pain and swelling and positive ANA, positivity for ANA. And she then developed lung disease, and this is very severe lung disease, interstitial lung disease in this case, or it can also be intra-alveolar hemorrhage. And she had a mutation in copper, which interestingly enough was inherited from a mother who at the age of 31 was completely normal. And to cut the story short, we did interferon signaling and we found high levels of interferon signaling. And I can tell you that copper syndrome is associated with a lot of abnormal interferon signaling. But I want to show you give you another clinical scenario now. So this is a girl, a 15-month-old girl born early, and she had she was thought to have lung disease that was due to the fact she was born early. But her mother had been diagnosed with rheumatoid factor positive polyarticular JRA, and then she developed lung disease, and her mother had been diagnosed with rheumatoid factor positive polyarticular JRA, and then she developed lung disease. So, I mean, I've put this up, I've said this is copper, and I have to say I thought that was copper, but actually that was STING gain of function. So there is a lot of overlap between STING, Savy syndrome, and copper syndrome. Having said that, we never see skin disease, or I've never seen skin disease in copper syndrome, but you don't have to have skin disease in STING in Savy. Okay, and just to say then, we can add another disease to have this kind of hand phenotype, copper syndrome. We've seen this kind of hand disease in copper syndrome. Okay, so a final kind of example that I just want to sort of flag up for a moment is that I've mentioned that, this is about a problem where you confuse self and non-self, self-nucleic acids with those of a virus. And essentially in the cell, there are only two sources of self-nucleic acid. One is the nucleus and one is the mitochondria. And I just want to mention the fact that mitochondria are stuffed full of DNA and RNA. And recently, there was this mitochondrial disorder, ATAD3A, that I talked about earlier on, that had been diagnosed in a neurological disorder and was said to be a classical mitochondrial cytopathy. But we came at it the other way. We referred these patients with a clinical and skin biopsy diagnosis of systemic sclerosis or scleroderma. And we did the interferon signaling, found that to be high. Then we went looking for the genes. And that's when we found that they also had mutations in ATAD3A. And the fact that someone had already described mutations in ATAD3A wasn't a problem for us, because actually we'd learned, by doing it kind of the wrong way around, we'd learned some very interesting biology. And I think essentially what's happening here is that the mitochondria are dysfunctional. You get a leakage of DNA out of the mitochondria, which stimulates the cytosolic receptors, cGAS, and then STING downstream of that. Okay, so I'm a clinician. I'm interested in treatments, and thinking about this kind of stuff takes you, as Dan has shown so beautifully, can possibly take you to treatments. And so one of the things we've been interested in is the idea that most of the genomes referred to as junk DNA, and that a considerable proportion, perhaps 40% of the human genome, is derived from ancient virus that we've incorporated into our own genome. So I really like this statement that the conceptual distinction between self and non-self breaks down when considering endogenous retro-elements. And essentially, we wondered whether endogenous retro-elements might be a source of self-derived nucleic acid that might be triggering a type 1 interferon response. And so we were interested, and you can make a case for some of these genotypes that I've mentioned very briefly in passing as in some way blocking the life cycle of retro elements as they go from a piece of DNA to a piece of RNA, and then this reverse transcription step back into DNA, double-stranded DNA, and then a kind of copy and paste into the human genome. So we did a clinical trial using reverse transcriptase inhibitors, which are used for HIV, an exogenous retrovirus, but here we were targeting endogenous retrovirus. And essentially, we think that we saw an effect of these reverse transcriptase inhibitors on interferon signaling during the period of the patients were taking the drugs. And we're trying to do a follow-up study to that initial pilot study now. But the other approach that we and others have been taking is to block downstream of the interferon receptor by using JAK1 inhibitors. And I briefly mentioned ruxolitinib already. So this is a little girl. And if I was with you, I'd maybe ask some of you, what's the diagnosis? And I think that some of you who, at least if any of you are pediatric rheumatologists, you might say she's got SAVI or STING, but actually that wouldn't be correct. What this child has is a D18N dominant negative mutation in TREX1, the same mutation as I showed you that picture of that Japanese woman with the deformed nose and the sort of affected cheeks. And this girl has had major problems with skin disease, particularly affecting the hands and the feet, and absolutely devastating disease for her. But this is her on day 0, and this is her 2.5 months after taking ruxolitinib. So clearly the ruxolitinib makes a difference. It's suggested that it's used. The rationale for using the drug was to block type 1 interferon signaling, but ruxolitinib, like baricitinib, like tofacitinib, doesn't just block type 1 interferon signaling. Ivona in the audience there, she reliably tells me that her dog takes ruxolitinib, or used to. It's apparently widely used in the pet world, and really it's used for an awful lot of things, seems to do an awful lot of things. So one can't claim the way it has an action here is specifically by blocking JAK1, but that's the reason why it was used. Other people, this is a very nice paper with a large group of patients with Aicardi-Gouttier syndrome from Adeline van Derve. But I want to, I come back to this idea, and most of you are not interested in brain disease, but that's where I started from. And I just want to tell you a little salutary and sad tale. So this is a child here with Aicardi-Gouttier syndrome due to biallelic mutations in RNase H2B. His brother was born, his brother was seen at the age of 4 months. At that time, the brother was unaffected, and we started on ruxolitinib, and he became symptomatic at age 14 months. So we, up until that point, we thought maybe that we'd cracked it, that, by treating early before we developed symptoms, that we'd sorted things out. That clearly wasn't the case. And what I want to draw your attention to is one of the challenges for the brain disease, at least, is that the levels of drug in the plasma are about 10 times the level of drug in the cerebrospinal fluid. And we need to get drug into the cerebrospinal fluid if we're going to address these brain disorders. More generally, I think we need rapid genetic diagnosis. I think we might need to use combination therapy. In terms of the brain disease, I think we might need to use intrathecal therapy. But what I would say is that this is a very exciting time in the world of these type 1 interferonopathies. And if these drugs come to be used and are seen to be effective, then all the more important to recognize a disorder as related to type 1 interferon signaling. But there are going to be drugs. The drug companies are very interested in making these kinds of anti-interferon drugs. And so I think the next few years will really see a blossoming of treatments in this area. But I want to sort of leave with a final note, which is that I think one needs to be, it would be naive to think that all of these disorders, some of which I've mentioned in passing, are due to just the type 1 interferon. And I think there's probably a spectrum. Most molecules, most proteins do more than one thing. They have, they don't just do the day job. And so there will be different diseases which respond differently to anti-interferon therapy. And I'm just going to end there by thanking all of the people and I won't name them, but the people in the lab in Edinburgh and the people in the lab in Paris. And thank you again for the opportunity to talk today. And congratulations, Dan. Thank you.